Hey guys. So yesterday we were talking about Watson and Crick's model for the structure of DNA, and how they noticed that it had a kind of, because of its structure, it suggested to them a way that it could replicate the unwinding and unzipping and then building the complementary strands. We call that semi-conservative replication, meaning half of it is conserved, right? Semi-conservative. Half of the molecule will be new and half will be old. And that was their suggested model. But we didn't know for sure if that was the case, if that was how DNA is replicated, until Meselson and Stahl came along. So let's look at their experiment. So they had a few different hypotheses, right? They didn't want to just go in thinking, this is what it's going to be like. This semi-conservative is what it's going to be like. They didn't want that to skew their results. So they thought, what are all the possible ways that you could replicate this helix, this ladder, twisted ladder shape of a molecule? And so they came up with a few different ways. And the most obvious one would be that you keep the old copy and you make a new copy just like it, an entirely new copy. And that's called the completely conservative or conservative model, where you conserve the original molecule. So one cell gets a strand of DNA that was the old one, and the other cell gets a strand of DNA that's entirely new before we do mitosis, right? Because this is happening in the S phase. So if you think about what that would look like, if you have a parental generation, well, I'm just coloring it black here. After one replication, right, you still have that original molecule, just like this one, and you have a copy that I've made in red. The copies are red, so new versions are red. And so it is completely conserved, so we call that the conservative model. After a second generation, right, this new copy would have to split to make two new copies, and we still have that original molecule from back here that's just been passed down through these cells. So in this second generation, one of these four cells still has the DNA, that original molecule of DNA, and the other three have an exact copy of that. So that's the conservative model. So the second model is this one suggested by Watson and Crick, this semi-conservative model. Semi meaning half, right? And conservative, kept or retained, right? Half of it is conserved. So here's our original parental molecule again. We go through S phase, and what happens is right down the middle, this unzips, and we build two new strands so that the new strands are here in red, and we still have half of each, right? We have each half of the original molecule appearing in those new molecules. And after a second generation, this would happen again. We're going to get a couple of molecules that are, right, these two right here, that are entirely new since the parental generation, right? Half generated here and half then generated in here. But so we've still got those original strands of the first generation's DNA. They're now stuck with half newly generated DNA strands, right? So it's semi-conservative all the way through. And then, for lack of a third hypothesis, they came up with this idea of dispersive, where the DNA gets broken into pieces, but then reassembled kind of randomly. And so you end up with partial fragments of new and partial fragments of old. And so they illustrated this kind of like this, showing that, the parental molecule is kind of scattered throughout each of its the copies of it, each of the new copies. And after a couple of generations, right, you could see we're just getting less and less of that parental one as it gets dispersed throughout more and more chromosomes as it gets copied again and again. So those are our three models. Like I said, that dispersive one is just kind of like to try and cover anything else they couldn't think of. So these are the three hypotheses. Bebo. Here's my cat, Bebo. These are the three hypotheses: conservative, semi-conservative, and dispersive. What Meselson and Stahl did was they grew bacteria on heavy nitrogen. So all of the nitrogen in the medium was nitrogen 15. So it had an extra neutron, it was heavier. And then they centrifuged that down and then gave them light nitrogen, nitrogen 14, medium. Bebo. She's just standing in my lap and purring. So what Meselson and Stahl did is they grew bacteria on heavy nitrogen, nitrogen 15. has an extra neutron to make it a little bit heavier. And then they centrifuged that, rinsed away that heavy nitrogen medium and gave them light nitrogen medium, and then let them grow for exactly 1 generation, did it again with another set of bacteria grown the same way, and let it grow for exactly 2 generations. So it's easy to do because E. coli, they grow about 20 minutes as a generation. So they did it after 20 minutes, they did it again after 40 minutes. And what they did is they isolated the DNA from those bacteria. So the parental DNA is grown on that heavy nitrogen, and it travels a certain distance when you put it in a gel column and centrifuge it, right? Heavier things are going to travel farther, lighter things will not travel as far. So let's look at the offspring, or the predicted offspring. We can make predictions for each of our 3 hypotheses. So if we look at the conservative model, you'll have 2 bands, right? Because we're going to have 2 different types of DNA molecules, the heavy ones and the light ones, after 1 generation. And that's what you see here. If the semi-conservative, there'll only be 1 band because each of the DNA molecules has basically the same mass. They are half heavy and half light. And so you get a single band somewhere in between. And the dispersive also is a lot like the semi-conservative because it works out to be about half and half for the first generation. Now at 2 generations, things get a little more different. The conservative still looks the same, but the semi-conservative is going to have that half and half band from those 2 molecules that have half old and half new, but you're also going to have those 2 molecules in our diagram before that were entirely new molecules. So they will be all light nitrogen. So they will not travel as far down the column. And so it will look like this with 2 bands. And the dispersive will have just a single band, but that band will be somewhere in between the 14 and the 15. Actually, it'll be a little closer to the 14 because the molecules are now like 3/4 light nitrogen and 1/4 heavy nitrogen. Bebo. This cat. Look at this. So beautiful. There you go. I know. Shh. Shh. So when we go to look at the actual data, here's the parental, right? So this is the test tube. This is the heavy end, right? The heavier stuff will go down here, and the lighter stuff will be closer to this end. So here's, actually, we've got it marked. Here's our nitrogen 14. Here's our nitrogen 15 DNA. My cat is very distracting. So let's look at what their data were, what they actually found. So after the first generation, this 20-minute one, we're trying to find the one that matches this. Well, it's got 1 band, and that 1 band is halfway in between the 14 and the 15. So that rules out our conservative model. It's definitely not that, because we would have seen 2 bands here, and they did not find that. After the second generation, they do see 2 bands. And so that, again, that locks us into that semi-conservative because they got one that's entirely new DNA, that nitrogen 14, that's this guy right here, and then they have one that is half and half. And that's the 2 different types of molecules. And if you go back and look at, a few slides ago when I was showing you the semi-conservative model, you can see where those 2 different types of DNA molecules come from. This is called the most beautiful experiment in biology. AP asks about it a ton. Everybody that teaches genetics asks about this a ton. It's a big deal. They ask questions. Sometimes they ask what would be the results if DNA replication had turned out to be completely conservative. And so you would have to say, well, we would have 2 separate bands because we would still have the original big molecule, and we would have the heavy, the nitrogen 15 molecule, and then we would also have new molecules that were the nitrogen 14. We would end up with 2 bands. Usually, they just ask to make sure you understand how it showed that it's semi-conservative, and it's these 2 bands that make it that clear. Some pictures of Meselson and Stahl. That's these 2 guys here. They're pretty cool, as you can see. Like I said, you should know that experiment pretty well. So Marshall Nuremberg did an interesting experiment. He figured out how to make RNA that was just a single nucleotide again and again and again. Uracil, uracil, uracil. Remember, RNA is not A, T, C, and G. It's A, U, C, and G, and that U is uracil. So he made an RNA molecule that was all U's all the way down, and then gave it to a ribosome to see what kind of protein it would make. And it turns out that the proteins made what are called polyphenylalanine, which is just phenylalanine, 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 phenylalanine. That was the chain of amino acids. And so he figured out that U's code for phenylalanine. And he started making new, he made all A's and things like that, and started figuring out all these different combinations. It's really interesting because he went to present this at a genetics conference, and the room was almost empty. It was in this little corner, maybe in the back of the conference in a little room. And he presented, but Crick was there. Francis Crick was there. And Francis Crick saw this presentation and was like, this is incredible. This guy just cracked the code. He just figured out how we get proteins from DNA. And because Crick realized how important that was, and Crick already had a huge reputation, he said, you're going to give your presentation again tomorrow in the big auditorium, and everyone's going to be there. And they were. So Nuremberg gave his presentation in front of the entire conference, basically, got a standing ovation. Everybody recognized the importance of his achievement. And so basically, we figured out this central dogma idea. Before we go through the central dogma, right, DNA, of course, can do replication. Does that in S phase, before mitosis or meiosis. But the central dogma is this idea that DNA is used to make an RNA copy, and that's called transcription. And that RNA copy is used to make a protein, and that's called translation. Right? And translation is done by the ribosome. Transcription is done by proteins inside the nucleus, of course, because the DNA is there. And we'll spend some more time on this. We will look at all three of these processes, replication, transcription, and translation, in a lot more detail over the next week or couple of weeks. So hope everything's going well with you guys. Hope you're not going too crazy. Hope these lectures don't suck too much. Miss you, and I'll see you. Shh, your purring is too noisy.